All right, that was a tough act to follow, but we have a very able group of people here, I think, that we can try to follow it with. And our charge was to discuss ensuring patient safety and innovation, and this is something near and dear to the hearts of, I think, all of you and certainly everybody on this panel. I don't know about the rest of you, but I spent most of this morning going, gee whiz. And I'm a gee whiz kind of guy, but the technology we're seeing, the advances in the science on a weekly basis don't cease to amaze me. You know, the circulating DNA stuff, I think, could be a real game changer, as well as the things we've seen in tissue. And so the whole point is, how do we ultimately get this into the clinic as quickly as we can, but as responsibly as we can? And I also want to give credit to many of the speakers this morning who weren't just saying, rush, 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 let's get this going, but almost every one of them said, let's make sure we do this in a responsible way. There are patients at the end of these discussions who will or will not benefit from our therapy. And one of my mantras in the diagnostics field, and I've been in this a long time, is that, and it's becoming increasingly more true, is that a bad tumor marker is as bad as a bad drug. We're starting to use these things even more so to direct how we treat patients, and therefore we need to be more careful and more certain of what we're doing. Dr. Varmus's talk, I thought, was terrific and really echoed this, which is that, again, I thought he was even going to say it, a bad tumor marker is as bad as a bad drug. In the old days, oncologists, and I am one, didn't really value markers very much. We ordered them and ignored them and treated people the way we thought, but increasingly that's not true. And so the whole point of the next hour and a half or so is to get to that. I just want to introduce my co-panelist. On my left is Ellen Siegel, who I think most of you know. She's the founder and chair of the Friends of Cancer Research, and she has led meetings like this in several venues that have frankly changed regulation and how we treat patients, so I give her credit for that, and supported a lot of the research that we've been trying to do. Who's next? Okay, so next to her is Tracy Bush. She's the Director and Global Coordinator of Roche Diagnostics, Diagnostic Regulatory Affairs. Next to her is Vincent Miller, and he's the Chief Medical Officer of Foundation Medicine. We heard a lot about their assay today. And then finally, but last but not least, is Liz Mansfield, who is the Director of Personalized Medicine Staff for CDRH at the FDA. And as Dr. Hamburg was leaving, she said, I'll leave the tough questions to Liz, so we'll have tough questions for Liz before we go. Okay, is there a problem? Many of you are either in companies or in academics and have sat through innumerable, endless meetings in which people are discussing problems that you think really don't exist. And I've been in academics for 30 years, and I've spent more time than I like on those kinds of committees. And I'd like to argue that there is a problem and that we're not wasting our time here. The first problem is that there are precious few cancer biomarker tests adopted for routine care. I've been a member of the American Society of Clinical Oncology Tumor Marker Guidelines panel, and I've also been a member of the National Cancer Center Network breast cancer panel, both of those for over a decade, 15 years now actually. And at least the ASCO tumor marker biomarker tests for colon and breast, those are the two we've approached mostly, have recommended precious few tumor biomarker tests that should be used to take care of routine care, in spite of the fact that if you open your journals on a weekly basis, you feel like you're opening the Biomarker of the Month Club magazine, whether it's the New England Journal or Lancet or JCO, so on and so forth. Second is that there are cancer biomarker tests that are used in the clinic that don't either have analytical validation, in other words, we're not even sure that they're done properly, or clinical utility, and by that I mean we're not sure that the use of that marker actually improves patient outcomes compared to not knowing the results of the marker at all. And that raises three important definitions, and I think most of you in this room know this, but we're going to use these terms, and I think it's important that we're all on the same page. These were established by EGAP about 4 or 5 years ago now, led by Steve Teutsch. They were principally talking about genetic tests of susceptibility of disease and not necessarily tumor biomarker tests, but I think they articulated what many of us in the field have been trying to say for 15 or 20 years and just hadn't quite gotten it out. The first of those is analytic validity. Does the assay accurately and reproducibly measure what you say? That seems intuitively obvious, but it is often not. And David Rim gave very nice examples of arguably the two most important markers in all of oncology, estrogen receptor and HER2, and the incredible variability in analytical validity of those two assays. The ASCO CAP guidelines have made an effort to try to standardize how those are done, and we've done a pretty good job of that, but it's still pretty scary. And for those of you who know the Ki-67 assay, which I thought David was going to get to next, he didn't. It's a disaster. And if any of you have friends or relatives and Ki-67 is being used to direct their care, at least in breast cancer, I think you're making a big mistake. The IHC assays for Ki-67 are really a mess. The second is clinical or biological validity, and that is, does the assay actually identify a biological difference or a clinical difference that may or may not be clinically useful? And that's just as it separate one group into two with statistical significance. That gets us promoted, it gets us grants. It's very good for those of us who are investigators. It also sets the stage to go on to the third, but it doesn't help us take care of patients. And the final is the term I used previously, which is clinical utility, and that is, do the results of the assay lead to a clinical decision that has been shown with high levels of evidence to improve outcomes? And while it's boring for me to read that to you, every word in that line is important because that's where the rubber meets the road. So let's talk about these two. And this summer I had the privilege of publishing a paper with several colleagues who represent several nodes in this vicious cycle. And this started to occur to me 10 or 15 years ago, and over the last 5 years really gelled as a part of several Institute of Medicine workshops that were held on tumor biomarker tests. And it seemed like everybody was talking about the part for which they're a stakeholder, but no one was talking about the whole vicious cycle. And that is, Dr. Varmus actually used this word this morning, it was music to my ears, that we don't value tumor biomarkers the way we ought to. And I'll say it again, a bad tumor biomarker test is as bad as a bad drug. Therefore, because they're poorly valued, there's poor reimbursement for them in the clinic, and that was brought up today. But there's also a very weak regulatory environment, and Dr. Hamburg, sitting here, the head of the FDA, said that. So again, music to my ears, that people are paying attention to this. There's been low funding and investment for tumor biomarker research because the payoff's been too low for entrepreneurs to really get into the field, and the regulatory environment's been so poor, nobody really understood the rules very well or felt they could get around the rules by going with other ways. At one of the Institute of Medicine workshops, there was a speaker representing venture capital, Miss Epstein. You're not in the audience, are you? I'm going to quote you if you are, regardless. Anyway, I gave this talk, and I thought, she spoke right after me, and I thought, well, she's going to blow me out of the water because I sound like a communist when I'm giving this talk. And she actually stood up and said, that was a beautiful talk. Venture capitalists are not asking for unregulated approaches. We're asking for consistent regulation. And she used the example of the stock market. Nobody would put money in the stock market if there weren't regulations about insider trading and things like that. And the same thing is true for what we do. There's traditionally been lower academic prestige, although I think that's changing very rapidly, so I may take that off this slide. But there's been a lower ability and incentive to conduct properly designed clinical trials. Dr. Varmus showed a few today. Frankly, he showed all but 4 that I know of, and he showed, I think, 4. So that's 8 trials that are designed to test the test, compared to how many hundreds of clinical trials in oncology to test new drugs. And I'm a therapeutic as well. I understand new drugs are important, but again, we really need to bring the levels of evidence of tests up to the same level we have for drugs. Therefore, we have lower levels of evidence on which to base our guidelines. Therefore, our guidelines are usually pretty crappy because we don't have good levels of evidence to make it. I can't tell you how many times we said, well, we think it's good, but we don't have the evidence to really say that. And therefore, doctors either order the test and ignore the results, or they don't order the test because they think they're no good, or they order tests that are no good and act on them and patients are mistreated, and therefore the marker utility is poorly valued. And I can go round and around and around. You've already heard about the Institute of Medicine Committee to discuss the evolution of translational omics. This was led by my colleague at the University of Michigan, Gil Oman. It was convened because of the fiasco at Duke that I'm not going to get into, and we did not want to get into that. We were not policemen. Rather, we got into how could patients have been put on a clinical trial using an assay that was not analytically validated or stable. And out of that discussion came this, and if you remember nothing else that I say, I think all of you who are developing new assays should remember this slide. This was made specific for the development of omics-based tests, but I think it's true for any diagnostic. In fact, it's probably true for any drug. It's Science 101, and that is you start out with a discovery phase, a good idea on the far left, and when you think you've really gone further, you go into a test validation phase where you actually make a test. Remember, a biomarker is not a test. A test is a test for the biomarker. HER2 is the biomarker. There are at least 8 or 9 different assays for HER2 out there. Once you have that locked down and you've developed clinical validity, then you cross the right line and say, are there data or do we need to generate data to demonstrate clinical utility? And there are basically 3 pathways to do so. Did I put that in? One is a so-called prospective retrospective study using archive specimens that have come from high levels of evidence type trials done previously. The prospective retrospective terminology was coined by Rich Simon in a paper that he and Soon Paik and I had the pleasure of writing about 3 or 4 years ago. And we set up a hierarchy for what kinds of studies would actually lead to level of evidence 1 studies. They're not the kind where you just pull samples out of a freezer, have lunch with a friend, put an assay together and get a p-value of 0.04 and rush it off the New England Journal and hope you get lucky. Rather, it's the kind, instead of saying, let's get an answer and then ask what the question was, preferably, let's ask the question and see if we get an answer that satisfies what we're trying to do. Thank you, Max. You taught me this about 20 years ago. If those kinds of archive specimens don't exist, then you must go into prospective trials. You can either do a prospective trial where the marker doesn't direct patient management. Those are usually to ask prognostic questions or monitoring questions, or prospective clinical trials where the marker does direct the patient management. And there are a number of publications that can give guidance. Dan Sargent wrote a brilliant one in JCO in 2005, and Lisa McShane and Bruce Friedland wrote another one in GNCI. They're almost identical papers that I would strongly urge you to read if you don't have them. So, okay, I forgot I put those on. There you go. All right, so in our paper, The Vicious Cycle, we made 5 broad recommendations. In fact, we got pretty specific, but one was to reform the FDA. None of these recommendations is set in stone. These were all just ideas we thought might at least lead to breaking that vicious cycle. And I have to say the one thing that we keep emphasizing is if we just fix one node on that cycle, we're in trouble. If we make it harder to get a biomarker test approved and we don't increase reimbursement, you all, those of you who are in industry, will walk. If we make it, reimbursement must go up, but we don't make it harder to get high levels of evidence for the test, the third-party payers will walk, and so on and so forth. For the FDA, we recommended that they should have a single oncology product line. So rather than having tumor biomarkers on the devices side and drugs on the therapeutic side, and we have ODAC, the Oncology Drug Advisory Committee, which really just looks at the drug side and or companion diagnostics, but they don't look at any of the other biomarker tests that we have. We recommended that there be an oncology product line, an OPAC that would look at all these things with corporate memory and expertise. This gets to what Dr. Hamburg was talking about a minute ago, the lack of expertise in the FDA with all these new things coming, and we can help them, I think. We recommended to reform enforcement of the discretion of LDTs. Frankly speaking, if I ran the world, we wouldn't have LDTs, or at least we would grandfather in the old ones but not have new ones. That's my own personal opinion here, though. And we heard Dr. Hamburg address that. Use analytical validity and clinical utility for approval and not clinical validity. Liz will, I think, either correct me or attack me, I'm not sure which, but on the most part, you've used clinical validity or whatever the manufacturer said the intended use was, and we'd argue it should be clinical utility, and that's what we've used in the ASCO tumor marker guidelines panel. And we would insist on a biospecimen bank being established for new drugs and tumor biomarker tests. This raises all kinds of legal and IP issues, but that's okay with me. I think we could come up with clever ways of doing this so that when a study's over, the so-called companion diagnostic, which is probably anachronistic by the time the trial's over, could be forgotten, and we'd actually find out what test really does help us decide which patients benefited and which didn't. We recommend reform of third-party reimbursement so that tumor biomarker tests are commensurate with value. That doesn't mean we overprice them the way we've overpriced drugs. The way we pay for drugs in this country is crazy. We don't want to reintroduce that, but we do want to have them commensurate if a good tumor biomarker test is as good as a good drug. We'd like to raise the level of funding to equal therapeutic research. We heard a lot about this morning. We need more of that. So the NCI has been great in a variety of settings helping us. We'd like them to be able to do more. We'd like to have the editors of the various journals actually adopt and enforce the various criteria, risk, remark, and the registry criteria. Right now they give it lip service, but they almost never actually enforce it. I've gotten tired of writing emails to my friends who are editors of the New England Journal and JCO saying, why didn't you insist on remark for that tumor bio? Oh, we forgot. Well, they need to quit forgetting because we got a lot of lousy papers out there, some of my own. And I don't want them to do it when I submit, but for everybody else. So. And finally, the guidelines should be evidence-based. And if we do all this, I think we'll have a virtuous cycle where tumor biomarker tests will actually be valued and therefore be used because we have high levels of evidence that they improve clinical outcomes. And I'll say it again, at the end of all this, there are patients who deserve this. This seems like a lot of more regulation. In fact, if anything, I think it will improve and enhance and speed up tumor biomarker acceptance because there'll be rules now. None of us liked the call, none of us who are Red Sox fans, 2 nights ago, but at least the rule was clear and everybody agreed that there was obstruction and the guy would have scored if he hadn't gotten tripped. Okay, I accept that. I'm a Red Sox fan, but we lost. The same thing is true here. I think we need better rules so that we have better biomarker tests to take better care of our patients. All right, having said that, to set the stage, I'd now like to introduce Ellen, who I've already told you who she is, and she's going to go through a lot of the things that the Friends of Cancer Research have been working on. Ellen?
Good afternoon. Thank you, Jan. So I just want to start with the obvious. Test and verify, get the right treatment to the right patient quickly and safely as possible. This seems obvious, but it's not so obvious. And it's happening, we heard earlier, it's happening every single day where patients are relying on these tests and we have no idea if they're valid. So we all know about personalized medicine, precision medicine. Recently, Friends, AACR, and ASCO convened a Hill Day, and there were about 90 cancer center directors, and I think maybe 30 or 40 of them told us about their assays and their tests. And I said, how do you know it works? And I didn't get a good answer. So I think we're relying on a lot of these tests that are not regulated, and we don't know if they do work, and we're depending on them because that's where the science is going. So these are huge issues because we're being guided by them, and one can hope that at least in a medical center where there is some level of validity, but in community settings this is happening also, and it's very frightening, and people think because they get a test that it works and it's accurate and it's going to guide a treatment. I think if we're going to do anything about this, we have to look at the regulatory environment, we have to look at reimbursement and policies that are needed, and possibly even legislation, because this is really important. Initially, I was going to talk about a companion diagnostics with breakthrough therapy. Over 75% of breakthrough therapy designations have utilized diagnostics. So we, along with the Alexandria, published a paper, and we worked with the entire community on this and had recommendations on how this can be expedited. But again, the basic principles were the same. They need to work, and nobody wants to rush through things that don't work and that are not safe and effective. The issue is what can be done in the beginning and what can be done in a post-market situation. I have to tell you, FDA was extraordinarily responsive. They were at the table in every step of the way with developers, with patient groups, and with academics. We worked on this collaboratively. And I was really worried that we were not going to get consensus, and there was enormous consensus on it. But so many of the recommendations in this publication, which you have outside, really can really be dealt with through not just on breakthroughs, because again, 75% are with breakthroughs are with the diagnostics, but we need to validate them. They're incredibly important. So we need to ensure that molecular diagnostics are working in clinical practice. I mean, so part of this is safety. Identifying patients for whose benefits outweigh risks And right now, this is the Wild West. I mean, we're starting to kind of put our arms around it, but it's not there yet. So tracking diagnostic tests in post-marketing settings is challenging, but it's moving in the right direction. So tests can be tracked with CPT coding, unique device UDI system rule finalized in September 2013. Well, it's going to take a long time for this to be there, but we really have to start to do this if we're going to really see if this information is right and if it's safe, and we have to know the tests that are being used out there. Right now, there's routine safety and effectiveness tracking through the FDA Sentinel program. We have the Reagan-Udall Foundation that has the iMeds program that really is looking at methodology, and I think we're going to be able to do a lot of things there with CER. It's just beginning. We're just getting it up and running. We're going to really work with PCORI on this. You heard earlier from Francis what's going on at PCORI. I think there is really an ability now with infrastructure and some directed funding to really figure this out. But at the end of the day, my message is very clear. Patients are relying on these tests. Treatment is being guided. And we need to know that they're effective, that they work, and that they have some validity. So this is a huge issue because I cannot tell you how many patients think because they get a test from their physician or from a lab that it's accurate. And there's a lot of information out there that is really inaccurate. And what Dan said in terms of biomarker validation or this whole vicious cycle is incredibly important. But as we start to get into 50% of our treatments are going to have these assays. Do they work is a very fundamental question, and should they be regulated? I say yes, but we need to figure out how that works, and yet without stopping innovation. Thank you.
So Tracy, you're up next.
Great, thanks. I'd like to thank the conference organizers for the invitation to speak. And I just wanted to spend a few minutes sharing with you some proposals for increased regulatory efficiency for the regulation of tumor biomarker tests that are becoming in vitro diagnostics, or IVDs. And again, we all need to keep in mind that the purpose of regulation is to ensure public health and patient safety. And so any proposal for increasing regulatory efficiency really must ensure that patient safety remains paramount, and that the existing standards of safety and efficacy for IVDs are upheld. And it's equally important to point out that test systems really must be held to the same evidentiary standards, regardless of the site of manufacture, whether that manufacture is being done by a commercial diagnostic company or by an individual laboratory. So one of the best ways to improve regulatory efficiency would be to adopt some sort of risk-based approach. There are a number of groups that have been working on this issue for a long time. Dr. Siegel just spoke about the Friends of Cancer Research Initiative for a risk-based proposal, and also AdvaMedDx has had a long-standing risk-based regulatory proposal that they've been working on. And there are a number of elements that are very common to both of those that are really important. A risk-based approach is preferable because this type of approach allows flexibility and encourages innovation without lowering the standards. So it really does keep the patient safety at the forefront. The risk-based proposal that I'm going to talk about a little bit really relies on the sponsor of the device determining what kinds of studies are appropriate for their product, and then requesting flexibility on certain requirements from the FDA. And this can be done by taking advantage of the FDA's pre-submission dialogue process, which has been used with great advantage with a lot of these companion diagnostics and other biomarkers. And the way this type of approach would work is the sponsor proposes flexibility for certain requirements and provides information to the FDA during the dialogue to justify the requests, and then the FDA would make a decision. And there are a lot of ways to look at risk-based proposals, but what I'm going to talk about here are proposals at the level of an individual device or tumor marker test that's being considered. And I'm going to look at proposals for increasing efficiency of analytical studies. Some examples of how to do this would be to ask the FDA to allow the use of contrived or artificial samples instead of needing to source actual patient specimens to do all of the necessary analytical validation work that goes into a registrational package for an IVD test. It can be very difficult to source these native patient samples, especially if you're looking at a tumor marker test for a rare disease type. So allowing the use of, say, spiked serum or artificial cell lines for demonstration of certain analytical performance characteristics is one way that this testing and the regulatory process could be made more efficient. Another possible proposal would be to allow the sponsor to use internal studies to add additional specimen types instead of having to do external clinical studies in order to add additional specimen types. There are a number of other possible proposals for increasing the efficiency of analytical studies, and the ones that make sense for an individual product are going to be up to the manufacturer to determine. And the details, of course, are going to depend on the specific assay technology that the test is based upon. We heard about a lot of new technologies this morning. And if you just look at what's available now in terms of IVDs for tumor biomarkers, there are tests based on immunoassay technology, there are tests based on real-time PCR technology and other molecular diagnostic technologies, and there are a lot of tissue marker-based tests that rely on FISH or IHC. And the details of analytical testing vary depending on the particular technology. So some additional proposals for increasing regulatory flexibility for tumor marker IVDs are listed here. One thing that becomes important, one of the proposals that Dr. Hayes made for demonstrating clinical utility is to do a prospective retrospective sample design. That can place a burden on the developer of a test because we are required to show that the storage of the sample did not impact the ability of the test to determine its analyte. And this can be really hard to do when the samples have been stored for a number of years and the test is only recently developed. So we could ask the FDA to allow for flexibility in terms of what evidence they would require the manufacturer to provide to demonstrate the sample stability in the cases where archive samples are used for clinical validation. Another idea that could be used touches upon multiplex assays. And again, we've heard that these are becoming more and more important in the area of cancer diagnostics. Instead of a system where you would need to do full analytical validation on each individual analyte contained in a multiplex assay, we could ask the FDA to allow the use of a representative sample subset to be used for certain analytical studies instead of having to do the full testing on each of them. Another thing that would be incredibly helpful would be to allow the use of a curated database to help establish clinical utility for rare mutations. And again, this can be important not only to test for an individual gene, like EGFR or BRAF, where there may be more than one mutation that's possible within that gene, but all the way up to next-generation sequencing types of approaches. We've heard from numerous speakers this morning about the difficulty in understanding the meaning of all the different pieces of information and mutations that a test may be able to detect. And the original standard of evidence for being able to have an assay approved for an individual mutation used to be that you had to demonstrate the clinical utility of that mutation in the actual clinical study for the device. And this, of course, becomes more and more difficult as more and more mutations are available. If there were a separate source of information that could be accepted as underscoring the clinical utility of an individual mutation, it would shorten the process of collecting that clinical evidence. And I think that would be a very big help in this field. Another thing that I think we're going to hear a little bit more about, and Dr. Hamburg certainly pointed out that this is a really important challenge for the agency right now, is to figure out how to regulate the next generation sequencing systems and to establish some kind of clear set of minimum criteria for the regulation of those systems. And this would include both issues of analytical validation and also how to demonstrate quality control of those systems, and also how to collect the proper clinical evidence. So these are just some of the ideas that would go into proposals to increase the regulatory flexibility and efficiency for regulation of IVD tumor biomarker tests.
And I think, what happened? Vince, I think you're next.
Yeah.
You have slides?
Yeah, I took the parallel approach.
Okay.
So I just have a few comments to make that really walk through how we at Foundation Medicine think about two of those three pillars that Dan pressed on, the analytic validity and the clinical utility, and really present both some of the challenges and the opportunities. So the number one piece is to take a patient-centric approach, and I think that's what everyone on this panel is talking about, and looking at that risk-benefit ratio in the context of where one is in the natural history of the disease and their particular cancer. And in most of the cases we see, of course, it's swayed toward giving the patient an opportunity, because more often than not, the prognosis is that of an advanced cancer patient who's received prior therapy. That being said, we felt driven to provide rigorous analytic validation of our approach, given the patient and doctor sitting at the other end of this, and having been in these conundrums many times in the clinic myself, seeing conflicting data perhaps come in. And so how does one approach, perhaps, and this does require a bridge from us to some others in the panel, but how could one approach validating a test that comprises 200 plus genes, of which you're sequencing the entire coding sequence of those genes, and in which your test simultaneously detects 4 major classes of DNA alterations, right? Base substitutions, small indels, copy number changes, and rearrangements. And so rather than slides, I provided my co-panelists with reading material, which was our Nature Biotech paper, which came out on EPUB last week, which really describes a 16 to 18 month collaborative effort with multiple academic institutions, including Memorial, using things including HapMap cell lines, other cell lines, orthogonal samples from multiple types of platforms, and which really at least proposes how one might go about validating a test with the type of numbers we as clinicians like to see. And I'll just quickly whip through it, but the essence is that for the base subs, 99% sensitivity down to a minor allele frequency of about 5%. For the small indels, and I think most of you are aware what the published algorithms around those are, and there we show 98% sensitivity down to a minor allele frequency of about 10%. For copy number changes, either homozygous deletion or copy number of 8 or more, 95% sensitivity. And parenthetically, the tumor purity becomes more of an issue around when one's looking for copy calls. And then finally, for fusions, correlation with well-matched series where there is an approved CDX, say something like the Alkvice's FISH test, or even a three-pronged approach with, say, IHC, which with the cell signaling antibody. So we did that. I think that's something that one can accomplish. We're big believers in there needing to be these types of standards. I'm sure there's many other approaches, many perhaps more creative approaches to doing this. And I think that's a necessary but perhaps not sufficient piece to getting us where we want for this type of testing. The next piece is something that we're all in together, and that's, I think, Dan touched on a key point, is I see my colleagues at Memorial ordering, getting their own test up and running, great test, and there's just a few guys who are raising their hand, saying we got to do studies to establish the clinical utility of this. And you saw 2 paradigms for that with the work David's doing with both the basket trial, but also the disease-specific concept. And that's something that Ellen and I have also been working on in parallel with the lung cancer master protocol around squamous cell carcinoma. But showing clinical utility for a test like this is not something that we grow up as medical oncologists thinking, boy, that's a really cool way to get a big paper or a grant, right? But yet it is, to some extent, it's the place to be right now. Because while it's implicit and intuitive to many of us, this is the way forward, the commitment to demonstrating this and to publish every case where, and both positive and negative, where we have an outcome, or to have those annotated in some ways that they're in the public domain is absolutely essential, so that the doctor who sees the PI3 kinase mutation in Kansas and wants to know if he should use try to get a TORC inhibitor off the shelf, will know that maybe in the XYZ database, those patients are 0 for 18 and he shouldn't do it. Or he'll be able to reach out to someone and say they're 6 for 18 and get context on that. So again, I think it's a little bit of reprogramming. I think we all know the components that's necessary to drive this. I would say about clinical trial design, one of the challenges, if you look at a broad-based test with many genes, the win in each disease may be very different. In, say, renal cell carcinoma, it may be showing that combinations of mutations of certain genes allow one to take a no-treatment approach. You have 2 cohorts of patients who have the same burden of disease anatomically, same functional status, and yet when you look at their genomics, you can see a clear path perhaps where observation works in one and not in the other. In contradistinction in a disease like lung cancer, we've seen that pie chart many times, and many in this room have helped identify slice in that pie. There one clearly can show multiple markers there with individual utility, significant publications, and even one could do an apples to apples comparison around cost of a test like this. And then there's other diseases where perhaps the linkage of the biomarker with the therapy, the targeted therapy right now, has been slower to develop, say in colorectal cancer, but where most of the markers may predict lack of efficacy of a class of drugs for which we test widely. So the one-size-fits-all clinical trial is, in my mind, incredibly hard to do, and one really needs to look at some of the approaches like the basket trial, like the master protocol, and then niche trials around diseases, thinking about the genomics of that disease, the standards of care, and how we might influence those. So I think I'll stop there and happy to field questions as they come up from Dan and others. Thank you.
And now Liz will tell us what the FDA plans to do.
Good afternoon, and thanks again to AACR and AdvaMed for inviting me to take the world on my shoulders here. So, yeah, I do feel a lot of pressure. I think throughout the day today we've heard about several new technologies that hold a tremendous amount of promise in the area of cancer treatment, whether it's diagnosis, application to therapeutics, or whatever. And I think what we've heard over and over and over again is the world is changing, and it's changing really quickly. And FDA has to change with that. We have to recognize that it is a new world, and we have to behave a little bit differently. And I think we understand that. Some of the things that I think the community needs to think about is how the new technologies will change how the results are viewed. For example, digital PCR we heard about this morning, and that is really a fantastic technology. But you do have to be careful when you think you need to use digital PCR in order to detect some actionable mutation in a tumor. Is really 1 cell out of 1 billion an actionable mutation? We actually don't know the answer to that yet. And so you really have to think about what is the context of the result here, and how does that affect therapy in ways that we can control. In fact, even with the old-fashioned companion diagnostics we have now, we don't know how much mutation you really need to have in a tumor in order to get a response. And we don't know whether more sensitive is better, because nobody's really studied that. So I think that there are actually a lot of big questions out there that we haven't even gotten to with the old technology, and now we've got new technology. So if any of you read the gray sheet, you probably will read that yesterday I said we were getting our co-development guidance finally together, and by the time it's published, it's going to be sort of overridden by all the new developments in diagnostics, which was sort of a throwaway remark and is not really true. The principles are still there. We still think that in the companion diagnostic, in the drug development arena, if you need a test to decide which drug you ought to take, that we think we need to look at that test, and because we have a lot of responsibility in determining the safety and effectiveness of the drug, and we need to know what the test is. On the other hand, the model that's been moving forward, as most people can recognize, is one test for each drug, is extremely clunky, becomes economically and sample-wise unviable when you've got to cut up the sample into 14 pieces to run all the different tests for the drugs that you might be able to take. Nobody can do that anymore, and insurance companies won't pay for all those tests, as far as I've heard. So I think the new technologies actually hold a tremendous amount of promise in kind of fixing this problem that we didn't really see coming. We didn't know everybody would jump on this bandwagon and be rushing to create drugs that had companion diagnostic necessary components. So what we're doing now is actually we've been working a couple years on looking at least the Nextgen sequencing area, trying to figure out, how does this technology work? What's the difference between the different platforms? And most importantly, what are the questions that we need to ask in order to satisfy ourselves and hopefully the patients and the physicians who are using them that the tests actually work? Well, that's a difficult question. And Francis Collins said, he thought everybody should have a whole genome sequence when their tumor was sequenced, which is really great. But we haven't even exactly figured out how, well, if you've got 3 billion base pairs, you're not going to analytically validate each and every base pair. I mean, that would be ridiculous, and we'd all be dead before you got through the first 10%. So we've had to come up with principles around what's an appropriate sampling of different kinds of changes you might expect to see, and so on for the whole genome area. But I would really be much happier if people didn't jump right into the whole genome and bit off what we might be able to chew now, which is something like a tumor panel. And I think there are multiple ways that we could move forward on this, one of which would be to have national tumor testing centers like they do in France. Dr. Varmus put up a slide where he showed all the information they got out of. There's like 12 centers in France. If you have cancer, your sample goes there, and each lab does exactly the same test, and they all have the same performance characteristics, and they collect all this information. Well, we don't do that now, and I'm not really sure we could pull it off. What we could do is to have, and again, I'm talking about next-gen sequencing, and that is not the whole solution. Some things can't be measured by sequencing at the moment. But let's just focus on that, because I think that's what people maybe are thinking about, is there's no reason why you can't have a panel of mutations that you think might be important. You can have a panel of 200, 500, 1,000, whatever. It's what you report that makes the difference, and it's what people do with those results that make the difference. So if you had a panel of 1,000 and you were only able to clinically validate or define clinical utility for 5, you could still test all those 1,000 and just report out 5, I think. I mean, there are plenty of tests out there now that measure more than they report. And then when something new comes along that you can define clinical validity or clinical utility for, you just start reporting that, and you've been testing it all along. So I think this is actually, we have in mind a model for an iterative process for adding things to next-gen sequencing as they become important for drugs, and so that you can take a single sample and get all the information you want. I asked a question earlier that maybe made a lot of people wonder what I was thinking, is what would happen if you tested for everything and then used the results to send patients to clinical trials? And some of the results you could treat off of because they've already been approved companion diagnostics, but to send patients to clinical trials, is that an attractive model? Is that something we need in our standard healthcare system now? Is to send more people with cancer into clinical trials so we get more medications with more evidence sooner. That's a model that we could consider, is, just go ahead and let everything out there. Then we have to trust people on the other end to use it correctly. In terms of what Tracy talked about, and manufacturers coming forward and proposing, what things they could do less of and what they might have to do more of depending on their test. We love that. We love people coming in with creative solutions. But what we really need is people to come in and talk to us. It's very hard to do this in a vacuum, and we're thrilled with the squamous cell lung protocol that's going on, because we're actually starting to get some of this stuff. We're actually getting to see how it works in real life, and there's some other protocols out there that are using Nextgen sequencing platforms and so on. And we need to see this, and we need to have proposals, and we need to talk to people about how this is going to be used in the community. We still have our standard of safe and effective, and do we need to, we can't change the law, only Congress can do that, but do we need to shift our lens on how we look at it? And in order to get the best information about that, we really need to be talking to people like you. So I encourage you to ask questions. I encourage you to come into us with proposals, or invite us to meetings, or do whatever you can, because really, as Dr. Hamburg mentioned, we can't have 100% expertise on everything. It certainly helps to be included in the conversation. Oh, and thank you for letting FDA have the last word.
You're the government. You're here to help us, Liz. We learned that a long time ago. By the way, I want to make one comment, and that one of our recommendations was this combined oncology products instead of an ODAC and the other. That is not in any way a slur on the expertise on the devices side. Liz and her predecessor, Steve Gutman, have taught me more than any school teacher ever taught me about this stuff, and have enormous expertise. Our feeling is that there is really sort of a lack of corporate memory, though, in terms of the people you do bring in from the outside, as opposed to a standing ODAC. And I wonder if you want to comment on that too, so that you have expertise in the clinical arena helping you, and that the therapeutic side has expertise in the devices arena.
The way that, I don't know if I'm on, can you hear me? Okay. Yeah, the way that we're working right now, at least in oncology, is we're steps away from Rick Pazder's outfit, and we all know each other by sight and by name. And so functionally, we're kind of operating as one unit now, although we still do have different laws, regulations, and ways of doing things that we are knitting together with various creative solutions and so on. And so I think that combining the two groups into one, you know, ab initio would have been a good idea. Trying to do it now would create some inefficiencies in regulation. But we do get a lot of expertise from them, and they get a lot from us. In fact, I think their level of consciousness about how much the test matters has gone up by orders of magnitude in the last couple of years since we've been working with them. So I think functionally we've kind of pulled off what you proposed without actually having to change structures and without rewriting the regs.
Ellen, you want to say something? Or?
Yeah, just briefly, about 10 years ago, many of us, ASCO, AACR, ACI friends, and others, worked with FDA for a cancer program at FDA, and some biologics were moved into CEDAR. And in fact, we've talked to FDA a lot about how that program is working, and it's okay. It's better than it has been, but it's kind of voluntary. But I think just because of the necessity and the complexity of what's going on, there's more collaboration. I think what may be lacking is more collaboration with the external community on it. But I think pretty much people are working together, and also with the NIH on these issues too, because they're very complex and different skill sets are needed.
I was also remiss. I told Ellen I might not feel good today because of the Red Sox game last night, depending on which way it went, and she told me her first boyfriend was Rico Petrocelli. Now, I don't know if any of you know who Rico Petrocelli was, but he's one of the greatest shortstops that ever lived, played for the Red Sox in the 60s. Well, I won't say when he played for the Reds. In the 90s, right, when you were in high school. So I've loved this woman for years, but now I'm completely enamored. So, David?
Yes, so I just want to bring up the question of clinical utility, because that worries me greatly. You know, there's a lot of talk that we don't want to inhibit innovation, but setting the bar of clinical utility is very high, in particular for rare cancers. So 50% of cancers are rare cancers. And showing clinical utility in rare diseases is pretty much impossible. This is something you can do in breast cancer and lung cancer, but it's extremely, extremely difficult for other tumor types. So I want you to comment on that, and I was hoping that Vince could comment on it as well, because they probably see a disproportionate amount of rare tumors sent to them, not just for decision making, but also for diagnosis.
Vince, you want to take it?
Sure. So in things we've presented before, about a third of our tumors are other, and sure there could be a few, misclassified or not classified perhaps properly, or the requisition came with a blank where there should have been some information. But that being said, it speaks exactly to your point that the oncologist in the community who's not a breast cancer or lung cancer expert is amidst his or her day seeing a salivary gland tumor, an adrenocortical tumor, a weird sarcoma, a skin appendage tumor, etc., non-colon GI cancer. How can one establish clinical utility for each of those diseases, which in and of themselves are almost certainly don't have a single or even oligogene unifying genomic profile? And so I don't necessarily have an answer for that, but I agree with David, it is exquisitely challenging. And perhaps there is a critical mass of a subset of diseases or a subset of genes that and a panel of experts can agree on that are relevant across multiple tumor types, or B2. Some people showed some nice slides of today would be a poster child, or TSC1 and 2. That's perhaps one way to think about this. Certainly, this is a little bit more of a technical issue. The ability to use a test like ours to potentially infer the site of origin of a tumor that's classified as an unknown primary is certainly a very robust hypothesis, and there's been some data on it with some other approaches that suggest it can be helpful perhaps in treatment decision making, and that's something we hope to report on our experience in the not too distant future.
I'll jump into that since I put the definitions up there, and I'll say it again, the words used in that definition were put there very carefully. For example, I didn't define what I meant by outcomes. So for breast cancer, which is what I do, we might say outcomes are improving survival, whereas for adenocarcinoma, or for cancers of the adrenal gland, you might say outcomes are responses, because you can measure responses easier and faster than you can, and perhaps a response in a patient with adrenal cell cancer is a meaningful outcome, and that would do it. The second is what is high levels of evidence, and Tracy, you raised, I think you used the term risk adapted or risk-based, and so again, for a marker for breast cancer, maybe you're going to have a higher level of evidence required to insert a test into clinical practice than you would for an adrenal cell cancer. I think these are things that, frankly, very reasonable and experienced people with experience in both testing and clinical, and it's important that they're both, and patients are all at the same table discussing these things. I agree with you, David. I'll say it again, I'm not a communist. In fact, I'm very entrepreneurial. But I think we do need rules. Otherwise, we don't have criteria for what we call success. And I don't care what the disease is, you still have to have some criteria for what you call success. I'll bring up again the CancerLink issue, Ask Us CancerLink. We have debated on the board of directors whether this could be the end of clinical trials as we know it. Will we just fade into an era of comparative effectiveness research where we collect data on hundreds of thousands, if not millions, of people with cancer who are treated, and look backwards and just see if we can figure out what happened? I'm still a clinical trialist. I still believe we need clinical trials, at least for most diseases, because of the science that's injected into a clinical trial, the rigor of determining what the outcome you defined ahead of time is and what a response is. I still believe in the scientific method that you need concurrent controls and not historical, but that's not always true. I challenge any of you to show me a randomized trial of cisplatin for testicular cancer, for example. It didn't happen, and that's because the historical controls were so compelling and the concurrent treatment was so compelling that if you do comparative effectiveness research, none of us would challenge that cisplatin cures testicular cancer. We've all hoped we'll get lucky like that. I happen to have been a med student in Indiana when Larry Einhorn was showing it worked, and it's why I'm an oncologist. I haven't seen it happen since. I think right now is an opportunity. We may start seeing this sort of thing. I hope that the stuff you're doing, David, at Memorial and other places will lead us there, but I still think we have to keep the scientific method in mind, but probably adapt it in different ways for different settings. And I think that's where we as leaders have to do this. We have to continue to be rigorous. I lived through the high-dose therapy and bone marrow transplant era for breast cancer, where we just kind of forgot to be rigorous. We just sort of said, gosh, it must work because it ought to work, and so it ought to work. And when we actually put it to the scientific method, it didn't. And so, again, I think it's a careful lesson we need to remember. Please.
Thanks, Dan. There may be entrepreneurial communists out there too now. This is a question for you, Liz, and I'm looking for an analogy, whether in a regulated product like a test or a device or a drug. You know, I was thinking about it when Francis and Peggy were talking, when it seemed to me that Francis was saying this is very fast moving, it's very tough to regulate in this. Peggy was sort of saying we have to find a way to, in this complexity, to regulate. So if you sequence a gene, it could be a cancer gene or it could be any gene, you will make occasionally some mistakes. And obviously we want to know about that. But you will also find areas that are difficult to sequence where there may be no calls generated by your calling algorithm on your machine. But more often than not, you're actually going to find new things, which we've already heard. We have to then decide whether they're benign or pathological. All that seems to me to be kind of the normal state of affairs and will be the normal state of affairs in cancer sequencing and other disease sequencing for a good piece of time to come. And that runs against the sort of traditional labeling, SOPs, you know, you make a mistake or you get something new, you have to pull it off the shelf and rejigger it, all that stuff. How is it, can you give me an analogy of a system that has evolved and is evolved, or in the past as sort of rapidly or as variably as what our knowledge about genomic sequencing is going to come out, and how the regulatory agency adapted to that? Well,
I would hazard to say I'm younger than you, so I may have missed a few years there. But actually, no, I don't think there's an analogy for the way information is coming at us so fast as it has from next-gen sequencing. I don't think there's an analogy for the technology improving so rapidly. What I do think is that diagnostic tests have never been perfect. That's not new, and that's not a requirement. Perfection's not a requirement. Truth in labeling, if any of you were around in the Steve Gutman years, that was Steve's mantra, is you can at least supply truth in labeling. Well, that would make me feel a whole lot better if I thought anybody read the label. So we still do ask for evidence, but we understand that when you look at a gene, you're going to find things you didn't know were there and that nobody's ever seen before. We understand that there are areas that are hard to sequence and that you can give an estimate for how well or how poorly you do that. So I think that's not necessarily news to us in the way that maybe people think that it is. And I tie that to the clinical utility discussion that was just going on. I think in some situations where things are very clear and we understand very well what the mechanisms of action are and so on, we can demand more information, whereas in a case of patients who have a cancer that has no standard of care, those patients are willing to take a chance on being treated based on some mutation that was found. And our responsibility and the device manufacturer's responsibility is to know that that mutation is really there, that that's not a false result and they're not throwing their lives into the hands of something on a bad test result. in a routine way. So no, I don't think this has any precedent. On the other hand, I think that a great deal of it is not new.
You know, Liz, this gets to the question David asked earlier this morning, and just a minute ago too, or was asking just a minute ago, and that again is that there's probably a risk-based assessment of who you use the marker in. So a marker that's used to direct care of a patient with ER-positive, HER2-negative breast cancer needs to be a lot more accurate than probably a marker for a patient for whom all available therapies have quit working and is considering the phase 1 trial. And I think it's very reasonable to have different criteria in those different settings because the stakes are different.
Yes.
And that's kind of what you just said, but again, David, I think you're concerned that I'm going to run around with a mask and steal all your exciting research away from you. To the contrary, I wish I was at Memorial. In fact, I emailed Dave Spriggs this morning and said, this guy, shut up. So let's take a question.
Right there.
Can you introduce? Oh, well, you don't have to introduce yourself, Andy.
Oh, I forgot to mention I went to Hopkins.
You know what? You're at Hopkins.
Hey, I went to Michigan.
I was just say, Tracy, where did you go?
So this is a follow-up question for Liz, actually to stay on the topic, but I'd certainly invite other panelists to comment if you have any observations on this. So continuing back on this question around the challenges of next-gen sequencing, what does it mean for regulation? I'm curious about this. Sort of seems like there's a metaphysical question at some point of like what exactly is the test. So in a world in which labs may be using a whole bunch of installed NGS platforms, and let's say a companion diagnostic developer comes to you, they don't actually have a test that they're going to market because presumably the diagnosing laboratory is using a platform they've already got in place. So presumably in that case, you're going to look for clinical validation of the markers, set of markers, and presumably that'll be in the drug label. But it seems to me in that case, there's really never any, or won't be, a physical test of some kind that really is coming to you for approval. And in the case of non-companion diagnostics, where you have at least some hook into the drug side, there's no test anymore. It's really just a query of the data set. I mean, especially if you run a genome sequence and you're looking for one thing, but 6 months later you decide to query that data set again, that's a test, but it's no longer something that I see how FDA can put its hands on at all. So I'm curious, if you can just put a finer point on how you might address that. I realize it's still a little theoretical, but it seems like a real question.
It is a little theoretical, and I disagree with you a little bit. I think every platform manufacturer's performance is different. Not that one is better than the other, they're just different. So I think at some level, every platform, we're going to need to see the fundamental performance of, but so it's a hard question because so much of how it performs depends on the informatics that comes at the end and has actually nothing to do with the actual process of generating the sequence. So in the end, I think the informatics, the sequencing itself may stay constant. We may have an idea, of what the sequencer itself has problems with and does well, but it's what the informatics does with the sequence.
And that informatics piece, that's a medical device, isn't it?
It is.
I should have said I assume that that's the scenario in which FDA probably has passed on the analytical validity of the platform, and I think you may have some applications for that, right? So I should have said I was assuming that in the first place, but it seems like then you get analytical and clinical validity are really separated.
So Andy, this gets to the IOM report, and actually Cheryl Nass is in the audience and herded all the cats into getting that figure made. We wouldn't have done it without her. And we really struggled with this. And when actually we said, we're not even going to talk about next-gen sequencing, because it seemed like a whole day we were talking about omics-based stuff and kind of ignored what was happening 2 or 3 years ago. But it's still going left to right, a good idea, and then some sort of platform that may not be the same if I sequence and you sequence, or even using the same platform if I use it and you use it. There's still errors being made there in one way or another. And that's got to be locked down before you cross that yellow line and start saying, should I use this to take care of patients or direct patients on clinical trials? And I know I'm speaking up when this is really Liz's question to answer, except I think in the IOM, we spent a lot of time in that committee, a lot of time addressing the very question you're raising, which is what's the difference between a biomarker, no matter whether it's a whole bunch of analytes together or it's one analyte, and an assay for the biomarker. And there may be, I saw you shaking your head yes when I talked about her too. I mean, there may be 8 different ways to measure that analyte, and they may not give you the same information. Please. Can you introduce yourself?
Felix Frew, Opus 3. This is sort of a follow-on to Paul's question, and then just a sort of a thought experiment, I guess. Why can't we look at sequencing, next-generation sequencing, as something more like an image, where you're looking at what you see in front of you, but you don't necessarily know always what exactly you are going to do with it? Because it seems to me that, and I think Francis Collins made that comment, that if we continue to look at individual biomarkers with an associated clinical utility for a specific purpose as it is defined in, let's say, a label, we're always limping behind. We'll never get to the point where we actually can use the information that we generate based on the latest and greatest information that we have access to. So can we get to a point where we're going to say, if we can analytically validate an assay, and we have a good notion about what it can be used for in the clinic, we could have a regulatory framework that acknowledges that status of a tool, let's say, and gives it the permission to be used in an environment where the information that's been generated with such an analytically validated tool is fed back into the network that really continues to create the clinical utility over time. Because if we don't do that, I'm afraid that we're going to have that very same discussion as we have today in 5 and 10 years from now.
Liz?
Yeah, so my answer to that is exactly. You know, there is no way of knowing how people are going to use sequencing or what they're going to find. So trying to force clinical validity on every single finding you would make in sequencing doesn't make any sense. And we understand that. Comparing it to imaging or radiology is an interesting analogy, and it's kind of true. The problem is we don't have people trained as whole genome sequencists. Radiologists know what they're looking for, they know what they're not looking for, and if you get 5 of them in the room, they won't all agree with each other when they're looking at an image. Is that a bigger problem in sequencing than it is in radiology? I don't really know, but I have a feeling that a lot more hangs on it. But in the end, yeah, you're right. We have no idea what people are going to use sequencing for, and in many cases, it will be used for things that don't have any clinical validity. And that's sort of what I was getting at this morning, is it's really what people do with the information that worries me. That's the part that FDA doesn't control.
Although as a clinician, the false positive rate on really increasingly more complex technical radiographs are a real bugaboo. I spend half my clinic day trying to figure out what's real and what's not. There's something there, but we don't know what it is. So I think, it's not that this doesn't, it's Diagnostics 101, in my opinion.
Right, yeah.
We have one more question and then we'll have to stop. Please identify yourself.
Hi, Patrick Bradley from Tapestry Networks. My question is for you, Tracy, although I think it might feed into something that Liz could speak to as well. You were mentioning with regard to the IVD registration, the risk-based approach, potentially using curated databases to show clinical utility. I'm a little bit confused because it's my understanding none of the current regulatory, the prevailing regulatory standards require the demonstration of clinical utility. So I'm wondering if perhaps we need a payer sort of on this panel, or if perhaps there are any ways to expand the regulatory purview to include that clinical utility determination, if you guys see that as a problem.
I think I may have been a little remiss in using the term clinical utility following Dr. Hayes' definition of the distinction between clinical validity and clinical utility. What I was referring to is the fact that today, to get a molecular PCR-based test on the market, the expectation is that we will have conducted a clinical trial that shows that we can detect that particular analytical mutation in a patient who is enrolled in a clinical trial for whom we get some clinical information supporting the intended use of the device, which if it's a companion diagnostic is related to a drug study and somehow related to outcome. And that may be a slightly different definition of clinical utility than the one Dr. Hayes was talking about. The problem comes when we've built a PCR-based test that can detect more than one mutation. Say it detects 6 mutations, but the clinical study that we did only contained patients that had 4 of the possible 6 clinical mutations that the test is analytically capable of detecting. In such circumstances, we've been required to develop software that masks the results of the 2 mutations that the test can pick up, but we didn't happen to get any of those patients enrolled in our clinical trials, so we weren't able to demonstrate clinically that the assay worked in those. And in a situation like that, which you could imagine would be magnified if you're using something like a multiplex panel or next generation sequence, it would be helpful to be able to refer to a database that says these 6 or however many mutations have clinical validity. The actionable clinical information, what to do when you find these mutations, is known. And so therefore, even if we didn't find all 6 of them in the study we did for the registration of our device, we'd still be able to produce and market a test that picks up all 6 of those mutations, because there would be this kind of database summarizing the clinical information for those mutations. Does that answer your question?
So with that, we've solved all the problems related to this panel, except for another 3 or 4 hours worth of discussion, another 3 or 4 years worth of discussion. I'd like to thank my co-panelists who I think have done a great job with a very difficult topic, and thank all of you for your questions and your attention. Thanks a lot.